Okay. So under immunity, we have presence of an antigen, okay, causes these lymphocytes to become active, your T cells and your B cells. Okay, T cells are usually activated first, causes the B cells to divide, and the B cells produce these antibodies. Okay, for now, we're going to call these antibodies these Y-shaped proteins. Okay. T cells, okay, they destroy directly via a chemical secretion. Okay, they destroy the antigen via a chemical secretion. Okay. Next thing. All right, so here's our antigen. Okay, so that's trying to denote the presence of an antigen. All right, the presence of the antigen causes an immune response. Okay, and this is going to go a couple different directions. Okay, one direction is called cell-mediated immunity. And the other direction is called antibody-mediated immunity. I'm just going to say MI for short for mediated immunity. Okay. So you have cell-mediated immunity, which is a bunch of soldiers that come out and do their job, and antibody-mediated immunity, which is different soldiers to come out and do their job. Okay. In cell-mediated, this is activation of the T cells. Okay, T cells are also known as T lymphocytes. Okay, so T cells, also known as T lymphocytes. And the antibody-mediated immunity, this activates the B cells, also known as B lymphocytes. Okay, so if you ever read this stuff or you see it on a test question or boards or whatever, T cells and B cells have those AKAs. All right, and then when this happens, they are in communication with each other. Okay, so T cells and B cells will communicate. Okay, when a T cell is active, it is going to destroy the antigen, the antigen by direct activation. Okay, so it destroys the antigen by what's known as direct activation. Okay. So I'll tell you my analogy in a second. Okay, when B cells are activated, okay, they have these antibodies on them. Okay, so those Y-shaped things are antibodies. Okay, those get released. Okay, so now these antibodies are free floating, so to speak, in your blood stream. So here's your blood tube, like your arteries. Okay, so it's released in blood. And these Y-shaped antibodies, they will go and bind to the antigen and destroy it. Okay, so that is known as antibody-mediated immunity, function of the B cells. Okay, my analogy is it's you. If you're a T cell and there's a bad guy, you are physically going to put your hands on the bad guy and subdue the bad guy. If you're a B cell, you are going to take your drones and send them out to go subdue the bad guy. Okay. All right. So let's talk a little bit more detail about antibody mediated immunity. Okay. All right. So antibody, I'm just going to do this shorthand, but please write it out. Antibody mediated immunity. Okay. So when you see that, okay, you want to be thinking your B cells. Okay, so it's activation of the B cells. Okay, now this next part is important. They're activated by a specific antigen. Okay, so they're for the most part highly specialized. Okay, so these B cells, once they're activated, they actually turn into plasma cells. Okay, and it's the plasma cells that release the protein called the antibody. Okay, so if you guys ever do reading, you know, with the immune stuff and you dive in and you start seeing, you know, plasma cells and, you know, all this stuff is, you know, it can be confusing. Okay, it's like, where the heck does this stuff come from? Okay.
Did you say that the B cell turns into a plasma cell?
Yeah, so once the B cell is activated, now that you've pissed it off and it's going to start wreaking havoc on the bad guy, that activation turns it into a plasma cell. And at that point, that now active plasma cell releases those antibodies.
Got it.
it's kind of like Tony Stark and Iron Man. Tony Stark by himself is the B cell, but once you piss him off and he's going to start wreaking havoc, he's going to get in the suit. So he turns into a plasma cell, and now he's got all the weapons and he can shoot those things and whatever.
My internet connection fell off a bit there. Can you scroll up a little bit to the second drawing that you did?
Right here?
Yeah.
Okay.
Sorry. Thank you.
You're welcome.
Okay. Got it.
Okay. Now this particular one, I'm also recording. I don't typically record all the lectures because I literally don't have the storage space, but this one I am. Okay, so next is B cell development. Okay, this one's kind of easy. It comes from the red marrow. So in other words, your bone marrow. Okay, remember you have red and yellow bone marrow. The yellow marrow is your fat, and the red marrow is what produces all the blood components. Okay, so the red marrow releases an immature lymphocyte. Okay, and then that eventually becomes a mature B cell. And yes, I'm skipping a lot of fancy mechanisms that we just don't need to know. Okay, it's fancier than that, but that's kind of the bottom line. Okay, an immature lymphocyte eventually matures into a B cell. Okay, then the B cell is released into the body. And it likes to hang out in blood and lymph organs. Okay, specifically like blood and lymph organs, such as your lymph nodes and your spleen. Okay, would be examples. Okay. Just for fun, I'm going to change colors. Okay. So B cells, or hold on, antibody production. All right, so B cells can divide. Okay, and this division it can turn into a plasma cell and a B memory cell. Okay. And the B memory cell, this is also part of the adaptive immunity where you learn how to fight something after your first exposure. Okay, the plasma cells, so there's the nucleus, and we've got the drones, also known as immunoglobulins. I made that up. That's not really an AKA, but just for our fun analogy here, These little drones are kind of hanging out inside the B cell, but once it becomes active, okay, now it turns into a plasma cell, and then these guys come out. Okay, and those are the antibodies. All right, the purpose of the B memory cells is for a quick response. Okay, a quick response to the same antigen. Okay. And these are thought to exist, depends on, I guess, the research and whatever. They exist in the body for years, decades, or lifetime. Okay. So I think it's a little bit all the above. I think, you know, once you get exposed, they do last for years, and they might could fade, certainly last for decades, and then might could fade, and then things just last for a lifetime. Okay. All right. Your antibodies are your immunoglobulins. Okay, so I'm going to be using the IG part. That's very common to see that, to read it, and whatever. Okay, so IG is an immunoglobulin, and these are your Y-shaped proteins. Okay, and they are divided into 5 categories. Okay, they're divided into 5 different parts. So we'll say divided into 5 parts. Okay, now I'm going to list these from most common to least common. So we have immunoglobulin G. Okay, that's the first one. Immunoglobulin M, immunoglobulin A, IgE, and IgD. Okay, so most common to least common in our body. Okay, by the way, you know, because of the times we are in, this is a clinical note, I do lab testing, lots of lab testing. You know, I draw the blood in office. We've got a centrifuge machine, all that stuff, and the different vacutainer tubes and so on. And we have a courier come pick up the specimens. Well, a common test that I'm ordering like crazy is COVID test. Okay. So there are 3, I mean, there's a bunch of ways, I guess, but the 3 that are being used right now, you do not have to write this down. This is just a rabbit trail clinical thing. One test is a NAA test. Okay, that's where you take the sterile Q-tip and you go way back into the nasal cavity area and you kind of twirl it around and try to get cells, and you send it in for testing. So I've got those kits. so we can do the NAA testing. And then the other test we can do is the IgG COVID test. And then the other one we can do is the IgM, okay? Now the difference between these two is if we want to know if you have the COVID now, then we're going to order the IgM COVID test. If you think you had it, so in other words, you had it, you're over it, you were symptomatic, all that stuff, but now it's good, which means it's in the past, then we're going to order IgG. Okay? So if IgG comes back positive, that means you had it. If IgM comes back positive, that means you currently have it. Okay? The NAA test is the nucleic acid amplification. Okay, that's what it stands for, nucleic acid amplification. And what we're looking for, I mean, think about that word, nucleic acid. We're looking for the DNA of viral cells. Okay, so that's why, or that's a test you can do. These two are the more common ones that I'm doing, that we're testing for. Okay. Okay, so these antibodies, also known as immunoglobulins, okay, they're a type of protein. They're found in the blood, okay, and other body fluid. Okay, we'll talk about that later. Their job is to, now keep in mind, you know, these are a type of Pac-Man, right? So their job is to neutralize the antigens, neutralize foreign bodies. They're designed to destroy bacteria and viruses. Okay. So, when you zoom in to an antibody, okay, it's the, you know, that Y-shaped protein, but when you zoom in, they kind of have these parts to them. Okay. So this part is the antigen binding site. Okay, so that's the antigen binding site. So what'll happen is, let's say this is the antigen. Okay, it will bind and begin the destructive process. Okay, that binding site, well, let me say that better. Okay, an epitope is the unique part of the antigen that the antibody recognizes. Okay, an epitope is the unique part of the antigen the antibody recognizes. Okay. All right, once they bind, okay, once they bind, it's called an induced fit. Okay, this allows for a very specific type of bonding. The induced fit allows for a specific type of bonding. Okay, and then that's one of the reasons for the unique, the memory, right? So once you develop these things and you have a memory B cell, and now you can do a quick response, those specific binding sites are now much more specific and much quicker because now you've developed them, okay? All right, we have four functions to the antibody process. Okay, so four. Okay, so four functions to the antibodies. In other words, how they work. Okay. Okay, so in step one, their job is to neutralize the antigen or antigens. Okay, in step two, or one of their second functions, they activate complement. Okay, so one way of looking at complement would be like asking for backup. Okay, so let's say you are fighting a perpetrator and clearly you're not winning, and so you're able to get on the phone and just call for backup, and all your friends and all these people show up to help you. Okay, that would be complement. So they activate complement, which means more help is coming. Okay, one of the ways they do that is through chemotaxis and inflammation. Okay. So as a fun analogy, chemotaxis would be like using the radio, a walkie-talkie to bring people in, and inflammation would be like using your phone. Okay, you're sending out a signal and people received the signal and they're coming in to help. All right. Third, it precipitates the antigens. Okay, precipitates the antigens, and I'll explain that in a minute. Okay, precipitating antigens, and then fourth, begins the phagocytosis process. Which means the actual destruction of. Okay. So on the right side of this, see if we can draw this out. Okay. So if we draw it out, neutralizing an antigen would look something like this, okay? So there's your antigen. There's your free-floating antibody through the bloodstream and all that. And then they bind. All right, so now we are neutralizing it. That is called the antigen-antibody complex. Okay, the second thing is now that we have the binding, okay, this activates the complement, and it's going to bring in white blood cells to the area. Okay. Okay, with the precipitating of the antigens, okay, so there's an antigen, there's one. Okay, what happens is these Y-shaped proteins, they start to bind to the antigens. So numerous antibodies bind to the same antigen. Okay, this is called a cross-link. Okay, and this one's my favorite. I love this one. The phagocytosis process, okay? So, we're looking at this, all right, but I'm just gonna draw one of them instead of the whole chain there. And now you'll see why I like my Pac-Man analogy. All right, the big eaters come in. The macrophage. Okay, so they come in and it literally eats this thing. Okay, so now it looks like this. Okay. Okay, what happens inside of the macrophage is the Y-shaped protein eventually breaks off. Okay, so now the antigen is by itself. The Y-shaped protein breaks off, the antigen is now by itself. Okay, and it is phagocytized. Okay, which means it's destroyed by lysosomes. Okay, now this next part, this is very important. And this begins, thus beginning the MHC, major histocompatibility complex. Very important. We'll talk about that later. Okay. Now, the way it does this, okay, if you look at the dot there, which is the antigen, okay. Now, visualize this. Visualize a like a cracker. Okay, you have a cracker on your desk, and you're going to take your fist and simply smash the cracker. What happens when you do that is the cracker is now busted into 1,000 pieces. So it's fragmented. Okay. So when the lysosomes come in to destroy the antigen, it is kind of like smashing your cracker, breaking it into 1,000 pieces. Okay, it is now degraded. Okay. Now what we're going to do is we're going to take a piece of that cracker. You're going to take one piece that you just broke apart, and you're going to bring it to the surface. Okay, you're going to bring it to the surface. All right, so now that particular piece of the cracker, or this broken down, degraded antigen, is now brought to the surface. Okay, now we'll get to that later because that gets into this major histocompatibility complex, and a bunch of fun stuff happens when this happens. Okay. All right, let's do one more thing and then we'll take a break. We're probably halfway through these particular longhand notes. All right, so we're gonna do one more thing, take a break, and that is, let's quickly talk about the different immunoglobulins. Okay, so we said that there were 5 of them. Okay, so gonna kind of just briefly list like what they do. So the IgGs provide the body with the most abundant we'll just say protection. Okay, so there's tons of them. They will go in and do that whole antigen antibody complex thing. And these are also the only immunoglobulin to cross the placenta barrier.
I'm sorry, what does that first word say? It says must something.
Oh, protection. So they're the most abundant, and their job is to protect you through this process here. Okay, the whole antigen antibody binding. Okay, another way of looking at this, if we can use a military analogy, you've got like 10,000 soldiers. Okay, you might only have like 2 airplanes and 5 tanks, which are the other immunoglobulins because there's not that many of them, but you got lots and lots and lots of infantry. Okay, and their job is to go out and stop the bad guys. Okay, and the IgGs also cross the placenta barrier. This is known as passive immunity. Okay, so active immunity is, you went out and, I mean, we're seeing it right now in Boulder at CU Denver, CU Boulder. And I don't know if you saw in the news or whatever, but there's a spike in COVID, because I mean, these are college kids, right? They're going to parties and fraternity parties and sorority parties, and they're making out and drinking and sharing drinks and all that stuff. So, and if they get sick, okay, they get sick, and then they'll probably fight it off, and that would be active immunity, right? So or acquired immunity. But if it's passive, so mom gave this stuff to baby, crosses the placenta. Another one where you see this that's really good is breastfeeding, right? The colostrum. Lots of good immune stuff in the colostrum. So those of you who might be doing like obstetrics, labor and delivery and all that, you know, I understand, I get it. At the end of the day, it's a personal choice, but if you can help it, you know, breastfeeding, especially the colostrum, is highly, highly recommended. Okay. And that doesn't last long. I think you only have like 3 days. So right after that baby comes out, you have like 3 days where that colostrum is going to be produced. And after that, that's it. Okay. And there's tons of awesome immune stuff and these immunoglobulins in the colostrum. One of the therapies I do with people is immunoglobulin therapy. Okay, so it is derived from colostrum. It's expensive, but people that are, you know, weak immune system, all busted up and whatever, we do it orally. So these are oral immunoglobulins that you take. They are derived from colostrum, and if you need something that's more powerful, then I send you out to immune people that do infusions. Okay. So if you're sick, but it's not brutal, we'll do oral. If it's just a mess and you're in a world of hurt, then we'll send out so they can do the infusions. Okay, the IgMs, these are expressed on the surface of B cells. And they're a very strong magnet, which means they have a high avidity. So they have a high avidity, which means it's a very strong bond. Okay, and it's designed to try to get this under control quickly. Okay, if you have a 100-pound perpetrator, you better get a 300-pound person to subdue. I mean, that is a strong bind. Okay, strong magnet. All right, IgAs. Okay, these are found in body fluids. Okay, such as your mucus. Okay, now write that stuff down. It's found in the mucus. It's found in your gut. Found in the respiratory system. Okay, it's found in the urogenital tracts. Urogenital tracts. It's found in saliva, tears, and breast milk. Okay, so kind of the fluids of the body. Mucus, gut, respiratory, the urogenital tracts, you know, how we pee and stuff like that. Saliva, tears, and breast milk. Okay, IgEs are the allergy. Okay, I'm emphasizing the way I spelled that on purpose because allergy, IgE, just a kind of a fun way to help you remember. Okay. It binds and triggers or activates a histamine response. Okay. If you've seen the movie Hitch, where he ate seafood and his face started to swell up, that is an allergy response. IgEs were definitely kicking in, and some other stuff. There's a bunch of things that are involved with an allergy response. And then in the film, if you saw it, he went later and got some Benadryl, which is an antihistamine, and in that case, it would help reduce the swelling, reduce the allergy response. And that medication reduces the allergy response by blocking the binding sites that cause the inflammation. So it kind of inhibits the histamines and all that from binding. So therefore, it's an antihistamine. Okay. So when you activate a histamine response, some of the other complement things that come in would be mast cells and basophils. Okay. IgEs are also known to help fight parasites. Okay, so if you're dealing with a parasite, the immunoglobulin E can come in and try to destroy that. Okay, the IgDs, they help as an antigen receptor. On the B cells. Okay, they will also activate basophils and mast cells. Okay, these are just other Pac-Mans, so to speak, that are coming in to help. Okay, now what happened, the reason why mast cells and basophils, okay, if you remember these from looking at them under a microscope in ANP2, they have granules in them. Okay, and when those granules are released, that would be like, I guess a bit of a stretch of an analogy. Be like pepper spraying someone. The stuff is contained, and then you release it, and then whatever it contacts, something's going to happen. Okay, so those are your granulated cells. All right. When we come back, we'll look at that major histocompatibility complex, and we'll look at how natural killer cells and all this stuff. Okay, so we got some fancy stuff coming up. Okay, so let's come back at 10:45.
I just had a quick question about you said the IgG test if you've ever had COVID and the IgM test if you have it now. Is that what you said?
Yeah.
So what if you get tested, like you get the test if you've ever had it, will it come up positive if like you have it right now?
Well, so if it's acute, which means you have it now, okay, so in a way, by definition here, it's a loose definition, but here it is. it takes about 14 days for the IgGs to show up. Okay. So if you have, let's say you have COVID like right now,
Mhm.
okay, and let's say you got it yesterday, you know, obviously it's going to take your body a little bit of time to develop the IgMs, but they are going to show up in the acute phase. Now, after about 14 days, then the IgGs come in. Okay. So IgM is if you have it now, and by definition within 14 days. And then IgG is if you think you had it. So if you thought you had it like right now, and I test you for IgG, it's probably going to come back negative because your body hasn't had enough time to turn this stuff into the IgGs. Okay, so it takes about 14 days for that to happen.
Okay, and then what about if you tested for an IgG, but you have COVID now? Would it come out positive or no? Or
Not
vice
if,
versa?
not if it's within the 14 days. Chances are it will come back negative if it's within that 14-day window.
Okay, because it has to grow or like
Right. Yep.
mature. Yeah.
Yes, it takes time to develop.
Okay.
All right. So see everyone in about 15. You and so that's fine. Like all this is within normal limits as your immune system is working. Okay. Hold on. Someone chatted in something. Yeah. Is it recording? I think I pushed start.
Yes, it's recording.
All right. Yeah. Thanks, Madison, for that heads up. Okay. All right. So I hope that helps with the mast cell. You know, it's in the white blood cell family. It comes from the myeloid group. Okay, we have the myeloid group and the lymphoid group. I think we're going to talk about that later. Look at it. I'm making this up, but look at it like it's neutrophil's cousin. Big cousin comes in. Mast cell. Okay. All right, now we're getting into major histocompatibility. Okay, major histocompatibility complex. Okay, so back at what we looked at earlier with the macrophage, okay, this big Pac-Man that comes in. Let's dive in a little bit more specific about that. There. Okay. All right, so as said before, the antigen antibody complex, it eats that. The antigen breaks off, and then we degrade it, which is kind of like smashing the cracker into 1,000 pieces. And then, you know, it does that by lysosomes. And then it's going to take one of these pieces. Okay, now I'm going to enlarge it. and moves it okay, it moves one of these pieces to the surface. Now let's make it a square. Okay, it moves one of those, now, and I enlarged that obviously, but it moves one of those pieces to the surface. Okay, this is now called a surface a cell surface protein. Okay, it's a cell surface protein. Okay, when that happens, okay, a signal hold on, I'm trying to change my colors. Okay, a signal is sent out okay, a signal is sent out and what happens is the cytotoxic T cells come in. Okay, so that activates or brings in the cytotoxic T cell. All right, so let's draw a T cell. Okay, there's the nucleus of the T cell. And what happens is it releases, okay, all these T cells that go into the site. Now, a cytotoxic T cell releases its drones, and those drones, I made that term up, okay, the drones. So these little drones are called killer cells, okay, so killer T cells. Okay, and they help to destroy the antigen. All right, the actual signal okay, the actual signal is because of interleukins. Okay. All right, it gets a little fancy, gets a little tricky here. Interleukins are cytokines. Okay. Cytokines are a protein that signal, that send out signals. Okay, so the word interleukin, okay, so inter means communication. Okay, communication. And leukin means produced by leukocytes. Okay, and your leukocytes, leuko is white, so these are your white blood cells. So they're part of the white blood cell family. Okay. And we have about 35 interleukins, and they name them IL-1 through IL-35. Okay, so interleukin 1 and interleukin 35. And guess what? every single interleukin has a very specific function. And we're going to know them and own them and test on it. I'm just kidding. That's too much. We're going to look at some of them. We're not going to look at all 35, but we'll definitely look at some of them so that you can see what they do and stuff like that. They're actually very interesting. Okay, so anyway, so we have 35 different interleukins. Okay. So we're gonna come back to this major histocompatibility stuff in a little bit. Okay. All right. Okay, remember, cell-mediated immunity involves the activation of T cells. Okay, so where do T cells come from? So earlier we looked at B cells. B cells basically come from the bone marrow. All right, and so where do T cells come from? Okay, so they come from the red marrow. And when they're in the red marrow, they are immature. T cells, also known as immature T lymphocytes. Okay, then they migrate to the thymus. Okay. Anybody remember the name of the hormone that the thymus releases?
Is it thymosin?
Yes, thymosin. Okay. So this activate, and let me say that better. This matures the T cells. Okay, so the T cells are not mature until they get to the thymus, and thymosin and all that fun stuff matures them. Okay, once they mature, then they are stored in your blood, lymph, tissue, and your lymph organs, such as your lymph nodes and your spleen. Okay, so T cell function. All right, we'll look at 3 of them. Okay, 3 major components of T cell function. All right, number 1 is the cytotoxic T cells. Okay, and these guys release lymphotoxin, and perforin. Okay, that's P-E-R-F-O-R-I-N, perforin. Okay, the lymphotoxin destroys the DNA of the pathogen. Okay, destroys the pathogen's DNA. The perforin causes the cell to lyse. Okay. And it does this by punching holes into the membrane. Okay, so pretend, we're fighting against a knight and he's got all the body armor on, that knights used to wear, and you kind of can't penetrate anything because there's armor. And then the perforin comes in and literally starts to eat away at the metal armor, creating holes. Now you can get in and start to destroy. Okay, the second one here is the helper T cell. Okay, the helper T cell releases interleukin number 2, so IL number 2. Remember, we have about 35 of these. So interleukin number 2, that is what stimulates the cell division of the T cells and the B cells. So let's say okay, stimulates cell division of the T cells and the B cells. Okay. So here's our helper T cell. And it releases the interleukin number 2. And it tells the helper T cell to divide. And the reason why is because it recruits more. I like the analogy with drones. It recruits more drones to fight and help. Okay. All right, the third one are the memory T cells. Okay, the memory T cells, let me think. Okay, these guys remain dormant. Okay, the memory T cells remain dormant until the exact same antigen presents itself. Okay. Kind of like saying, you know, oh, it's that guy, you know, don't be that person, right? So it's a very specific person typically when we say that. So they remain dormant until that particular antigen is present. Okay. And when it is, the memory cells, they're stimulated. Okay, so when the memory cells are activated, they convert themselves into cytotoxic T cells. Okay, remember, I think our fun analogy with that was, you know, you're Tony Stark, which means you're just you're a T cell that's just kind of chilling. And then the bad guys are coming in and you're going to suit up. And once you suit up, now you're a cytotoxic T cell. You're ready to wreak havoc. Okay, now this one develops after the first exposure. Okay, meaning the first exposure of the antigen. All right. So if you've never seen, okay, this is kind of a fun way, a fun analogy here. So just kind of roll with it. So let's say you've never seen virus number 1. Okay. So fine. Virus number 1 comes in and you're symptomatic and your immune system is fighting and then all that stuff. And eventually you get over it and eventually you create these memory T cells. Down the road, when you see virus number 1 again, no problem. Now you've got these dormant memory cells and now they're activated and now they've been activated and became cytotoxic T cells and they're going to destroy virus number 1 and you're going to be fine and you're probably never going to even know it. Okay. I mean, think about that. Our body is constantly, every millisecond of every single day is constantly fighting the bad guys and you don't even know it. Now, the only time you do know it is when enough stuff has been recruited to promote inflammation and promote the fever and the aches and the chills and all that stuff, which we'll actually look at that in a little bit. Okay, what causes that? All right. Okay, let's look at natural killer cells. Okay, natural killer cells. Okay, first of all, they are considered part of your innate immunity. These guys are cool. Natural killer cells they're crazy. They're so cool. Okay. Now what that means is you just have them. You're born with them. Okay. They're in your system. They're inside of you and they're working. Okay. The reason why they are called natural killer cells is because they do not require to be activated in order to kill cells. Okay, so they do not require activation in order to kill cells that are missing the self markers of the major histocompatibility complex. Okay, I'll explain that in a minute. So they do not require activation in order to kill cells that are missing the self markers of major histocompatibility complex. Okay. Slight tangent, and we'll come back to it. If you're like, what the heck is a major histocompatibility complex? All right, here's the easy version. Your body recognizes you from not you. Your body has the ability to recognize you versus not you. Okay, here's another way of saying that. Your body knows self from non-self. Okay, your body knows self from non-self. All right, here's an example. If I take your own blood out of you and then put it back into you, you're going to be fine. No rejection. If I take someone else's tissue and I put it in you, that's technically foreign tissue, which is non-self, and your body will destroy it. Okay, that's major histocompatibility complex. Your body's ability to detect self from non-self. All right. So if we do not do the appropriate testing and all the meds that come with it, and I just give you my kidney, your body's going to reject it. Okay. If you need a kidney and there's no testing and whatever, and I just give you my kidney, your body is going to reject it because it recognizes self from non-self. Okay, so that's what we're going to build on here with major histocompatibility complex. All right, so natural killer cells. Let's see, they also, let's see. Okay, they can destroy cancer cells. Okay, hold on a second. Someone chatted me something. They said, did you say natural killer cells are part of T cell function? No, they're kind of their own thing. Okay, natural killer cells are their own thing. Okay. All right. Just reading, make sure I didn't miss any more chat stuff. All right, let me get rid of that.
Are NK cells affected with chronic issues?
You mean like if a person has a chronic illness?
Mm-hmm.
Are the natural killer cells affected?
Yeah.
Do they maybe just stop working at some point?
Yeah.
Overall, probably, with, chromosome issues and other genetic problems and just things falling apart. Sure, I'm sure that's a thing. In general, not really. You know, if a person becomes immune compromised enough, yes, they stop working. But I would say outside of unusual circumstances, they're always working. But yes, certainly at some point, for whatever bizarre reason, yes, they can stop working.
Okay, I guess I'm just asking because they kill cancer cells, so people still get cancer. So what's happening to the NK cells?
Well, it's not so much that they're not working, it's like they can't keep up. You know, if
Oh,
you're
okay.
if you're the only soldier, you know, you could probably handle 2 or 3 soldiers coming at you at a time, but then you got 10,000 coming at you. It's just you're overwhelmed. You know,
Gotcha.
they just can't keep up. I mean, you know, there's limitations to the human body. You know, like here's an example. You know, if you get bit or if you get stung by a bee, you know, you've got venom in you, and in general, it's not really going to cause any problems other than local swelling. It's annoying. It's stupid. The pain goes away probably after 30 minutes and whatever. Okay. And then I think it's the black mamba. Okay. One of the most poisonous snakes in the world. Yeah. Get bit by that thing. See what happens. You know, you could argue, well, it's poison, just like the bee venom, but it's just a whole different level. It's so much, you know, your body just can't handle it. And it usually kills people. You know, another bizarre one is the box jellyfish. And that stupid thing, it's fatal. I mean, that thing kills people within like 5 minutes or something like that. It's like, what the heck? You know? So anyway, I'm just saying there's limitations to matter. And our bodies are pretty good at fighting off stuff. But at some point, whatever it is, being cancer cells or toxins or whatever, you know, it can just kill us. You know, it can really make us sick and kill us. I know some of that medical stuff, I mean, it blows your mind. Like, I'm not kidding. Here's an example. This stuff really happened. So a person will be in a high-speed motor vehicle accident. I mean, we're talking 150 miles per hour between the forces of the two vehicles. The car was destroyed, flipped over, rolled down a ravine, hit every tree on the way down, and the person was fine. No broken bones, maybe like a little abrasion here and there, but they were freaking fine. Right? And then you've got these little fender benders, and the person had like a carotid artery dissection and died in the vehicle. 5 mile per hour. I mean, crazy stuff. Right? You have all these exceptions to the rule, and I hear about it all the time from some of my medical friends. And they're like, yeah, we got this person that comes in, you know, and they talk, you know, struck 47 times by lightning, and they're fine. I'm exaggerating, but you get the idea. So yeah, those are exceptions to the rule, but you always have those bizarre opposite medical things, you know, super brutal, but the person's fine, super minor, and it frickin' killed him. Okay. Those things happen.
I've got a question for you.
Okay.
All right. So earlier you're talking about the cytotoxic T cells, and then they come in and they release the killer T cells. Are the killer T cells going to be the lymphotoxin and the peripherin?
Yes.
Okay.
Yes, absolutely.
Perfect.
Yeah, that's some potent stuff. That's good stuff right there. Okay, so let's hold on, where are we at? Can destroy cancer cells. Okay. So cytokines help recruit or I shouldn't say activate. That's the wrong term here. They help recruit these natural killer cells. Okay. Cytokines tend to respond to viral infections. Okay, the presence of a virus is actually what they consider its stress to the tissue. Okay, and this particular stress to the tissue starts to release chemicals or signals. And so then, you know, your Pac-Mans will come in to that site. Okay, anytime you send out a signal, help is on the way. Interferons also recruit your natural killer cells. And I think we'll talk about interferons in a little bit. Okay, so natural killer cells are produced in the bone marrow. Okay, so they do have the ability to determine if a cell is infected or not. Okay, is a cell infected? If it is, they will destroy it. If the cell is okay, then they leave it alone. Okay, and they do this. All right, so the natural killer cells can control their cytotoxic activity because of two receptors. Okay, so in other words, these natural killer cells have two receptors. All right, one receptor is an activating receptor, and the other receptor is an inhibitory receptor. Okay, so if activated, it will kill the antigen. If inhibited, that means it recognized self from non-self. Okay, and this is part of major histocompatibility complex 1. Self from non-self. Okay, we have 3 different major histocompatibility complex. Okay, so there's 3 classes of them. Class 1 involves the natural killer cells. Okay, so that's why your own body does not, let me say that better. Your body does not know, hold on, let me try that again. That's how your body knows to not destroy your own tissue. Okay, this is how your body, your immune system knows not to destroy your own tissue. By the way, what's that called if your body starts to destroy itself?
Autoimmune?
Yes. Autoimmune is all this, you know, normal stuff we're talking about is out of control. You're not, you know, in the military terms, it would be called friendly fire. Okay. So, I mean, that you don't want friendly fire, which means, you know, if you're all are soldiers, you're killing each other, and that's, you don't want that. You want to kill the bad guys. So our immune system, it just knows to leave you alone. But when that spins out of control, that is a very unfortunate thing. And that's, yeah, considered autoimmune. Okay. So let's draw a picture of how this works. Okay. So here's, yeah, that's terrible. Okay, there's tissue. So we'll say self. And here is tissue or cells or whatever that is non-self. Okay. All right, so the natural killer cell is going to come in, and it has these receptors. Okay. And one is the activating receptor, and one is the inhibitory receptor. Okay, tissue has those surface protein markers. Okay, whoops, yeah, that's good. Okay, so tissue has these surface protein markers, and this marker would be considered major histocompatibility class 1, which means self. So all of your cells and all of your tissue and all that inside of you has these special markers that says it's you. Okay, that's how your body can recognize self from non-self. Okay, so these natural killer cells are constantly scanning your body, constantly scanning the cells, constantly scanning tissue. So when the inhibitory part recognizes self, nothing happens. Okay, everything's okay. So it's inhibited. However, when the natural killer cell comes across and there is no self, as you can tell, self is not there, okay, then the activating receptor is activated, and it's going to release the perforin. Okay, it's going to release the perforin. Okay, and these granules that are released are going to lead to cell death. Okay, the granules that are released are going to lead to cell death. Okay, so you want this for tissue that's not yours. Antigens, foreign tissue, whatever. Okay. All right. Now let's look at the 3 different classes of the major histocompatibility complex. Okay. So with MHC, there are 3 classes. Okay. Class 1, Class 2, and Class 3. All right. Okay, these 2, Class 1 and 2, kind of go together. So that's why I drew the box around them like that. Okay, Class 1 and Class 2 are also known as the HLAs. Human leukocyte antigen. Okay, human leukocyte antigen. Okay, and class 3s are different various immune proteins, or we can say other immune proteins. Okay. MHCs, so major histocompatibility, are from a set of genes in chromosome number 6. So let's draw out kind of our version of a chromosome. So you have all right, so there's our chromosome. Okay. And let's just say about right there is chromosome number 6. And that's where these class 1, class 2, and class 3 come from. Okay, so it's a genetic thing. We have them. Every once in a while, there's a person that has a messed up chromosome number 6, which means they're going to have a messed up, MHC and HLA process, which means, chances are they're more susceptible to autoimmune problems and other problems. Okay. That gets into some really fancy testing, but it's testable. They can test for chromosome number 6 to see what is going on. Okay. Class 1 and 2 are part of that HLA. Okay, and the class 3 are those other proteins, immune proteins. Okay, the function, the HLA function is to present different protein fragments called peptides at the surface of the cell. Okay, and this is what recognizes self from non-self. Remember, You know, one of the things that we drew a while back was the macrophage, and you know, the ability to take the protein and become a cell surface protein and all that. Okay. Well, your body does that with you, right? It just makes sure that on the surface of the cell, there's a protein there that says, this is me. Okay.
Is that function for 1 and 2?
I'm going to get to 2 in just a second. Okay. Number 2 happens to get into, we'll get that in a second. Okay. Okay. So when this happens, healthy cells are ignored. Okay. And foreign cells, or you could say foreign protein, are destroyed. Okay. With the class 2, okay, these are the receptors found on the antigen presenting cell. So the APC, the antigen presenting cells, like on the macrophages and B cells. Okay. The antigen presenting cell alerts or activates the immune system. And when that happens, that activates a specific immune response. Which means it ultimately ends in the destruction of foreign tissue or a foreign protein. Okay, and then the class 3, would be complement. So these immune proteins would be things like complement and cytokines. Okay, so these are helpers that come in to help destroy if the histo, which means the tissue, histocompatibility, is this tissue compatible with you? That's histocompatibility. All right. One more thing. Hold on a sec. Okay. Well, let's see what we can get through, and then we'll break. A few more things. Okay. So we have draw a picture of the T cell. Okay, so there's that antigen. We broke it down. Okay, take it to the surface. There it is. Okay, the T cells will help with this process with the activation of a specific immune response. Okay, so once we activate help, an example would be T cells. Okay, so this T cell would come in, and these two sites would bind. Okay, and when that happens, it will release the chemicals that will help destroy the foreign protein. Okay, hang in there, we're almost done. Okay, cytokines. Okay, some examples would be interleukins. Leukotrienes. And interferons. Okay. So interleukins, we have interleukin 1 through 35. Okay. Leukotrienes. Leuko means white. Trienes means 3. Okay. And what they do is they bring in neutrophils. Okay. So neutrophils are part of the white blood cell, the most abundant of the 5. Remember the neutrophils, eosinophils, and basophils, and all that. Okay, the leukotrienes also help sustain the inflammation. Okay. And they can also cause, they help with the allergy response. Okay. So, increased mucus production, increased inflammation of the cells, all that kind of thing. So if a person has asthma, okay, this can spin out of control and cause decreased airflow. Okay, so they have a hard time breathing. So for asthmatics, they're a little bit more sensitive to being sick. All these things that we're describing here, you want these things to work, right? If bad guys come in your body, you want these guys to work. However, if you have a pre-existing, it can take a normal situation and kind of spin it out of control. So an asthmatic would be an example. Okay, let's mention a few interleukins. So interleukin 1 they tell the B cells to divide. Okay. They promote fever. Okay, so they're responsible for why you get a fever. Okay, and this is not an exhaustive list, okay? Where I mean these things are fancy. And what blows my mind is how they can figure this stuff out. It's kind of cool, that technology to figure this stuff out. Okay, interleukin 2. Okay, this one activates or recruits or brings in the T cells. Okay, these bring in the big guns, the B cells, and the natural killer cells. Okay, so if interleukin 2s are activated, okay, remember it's a type of cytokine, then these big guns come in. All right, interleukin 3, okay, it brings in or activates the T helper, the mast cells, and by the way, all of these natural killer cells are recruited by all of these, okay? So, you know, we could write it down 35 times, but natural killer cells, I mean, they're just always there. So they're always recruited. Okay, and as mentioned earlier, the mast cells are responsible for the histamine response. Okay, and then interleukin 4 are the CD4 cells. The mast cells. And then a lot of white blood cells like macrophages, B cells, T cells. So it brings in a lot of stuff. Okay, now as you can tell, the more that things become activated, the more the recruitment, the more powerful the response. That's why at some point, I mean, you know, as far as signs and symptoms, it just kicks your butt. I mean, there's one thing being a little bit sick. You know, yeah, I got little sniffles, little runny nose. And then, I mean, it's just miserable and in bed, you know, aches and fevers and chills and all that stuff. Okay. The CD4s, they're just another type of Pac-Man. Okay. And I'm not gonna, I mean, they're actually quite complicated how they work. Not gonna get into all that. Just know that it's a type of Pac-Man that comes in. Okay, almost done, like a couple more minutes. Interferons. Okay, the word kind of gives it away. They interfere with viral replication. Okay, they interfere with viral replication. They're kind of big guns because they go after viruses, bacteria, parasites, and tumor cells. Okay. The interferons are responsible for the muscle aches. And fever. Interferons are also used for some cancer treatments. Okay. Like prostate interferon pellets, for example. They'll take these interferon pellets and kind of embed them into the prostate and, you know, hope they do their thing. So with some modern, you know, we'll just say cancer fighting and, you know, illness fighting stuff, that's one of the treatments that, that they're working on and they can do. And the technology is just getting better as time goes by. All right. Okay, any questions now that our brains are fried? All right. So let's come back at 1 o'clock.
I had a question before we go to lunch regarding homework. I had some technical difficulties before I logged in, so I was like 15 minutes late this morning.
Okay.
Is there a Dropbox for the homework we had?
So the format, I mean, the way that I tried to post it on Canvas, isn't there a way to just kind of send it back as an email?
I believe so.
Yeah.
No, because then it's not connected to our calendar, so we don't know when stuff is due.
Right.
So there's a place where in assignments you can directly say like week 2 homework assignment, and then there's a submit tab and we submit it right there.
Okay. On the calendar, I posted all that stuff on the hold on 1 second.
Sometimes you have to hit publish in order for us to see it. I don't know if you have that on your end or not, but I know a lot of teachers are like, oh, I forgot to like make it active.
Is this the only spot? Yeah. Okay. So I'm sorry I missed that. Someone actually came to the door. So when you click on the calendar, do you all see the pathology? You know, I posted like when everything is due for the entire quarter.
It doesn't show to us.
If you let one of us have control, we can show you what we see on our calendar.
What do you mean let you have control?
Like to screen share.
Oh. How do you do that?
I think you just have to get out of your own screen share and then someone can screen share theirs. But since you're screen sharing, somebody else can't do it.
Okay.
You have to make somebody else the host.
Okay. Well, I have an appointment, an online appointment with Jen. Okay. So I think every one of you probably 300 times over sent her emails. So we are, you know, I'm scheduled to do an online thing with her.
This